So we are going to start our immunity unit, so our immune system unit. And so for a lot of this, we're going to have to talk about some vocabulary. So that's why your notes include both a glossary, which is your kind of quick reference guide for how to define things and any special features and things like that. And also it includes your guided notes. So just be aware that you might be flipping back and forth as we go through between the glossary and this to kind of fill in both sections. And I'll try and remind you, hey, this is something you'll want to add to your glossary, or this is a good time to open your glossary while we're going through the guided notes. So the first thing that we need to understand about the immune system is, what is the immune system even trying to defend against? And we use the term pathogens to describe a substance that causes disease. So most often we think about pathogens in terms of either cellular things, so these would be things like parasites or protozoa, fungi or bacteria, a prokaryote, or acellular, non-living things, things like a virus or something we call a prion, which is a detrimental protein. So if you want to flip to your glossary on the third page there, the second one says pathogen. So the function is something called an infectious agent. So an infection agent would also work. So why are bacteria bad? Why do we care if we get an infection with bacteria? Well, the bacteria kind of acts as like a microscopic parasite almost. It pulls nutrients from the host cell, and then it produces waste products. And then those waste products might be toxic to the cell, or the cell just has to use energy to get rid of those waste products. So it can be really detrimental to the host cell to not only lose nutrients and energy to this bacterium, but also now it has to deal with all of the toxins and waste, and those can cause damage to our tissues. Also, immune responses can also cause damage to cells. And we'll talk a little bit about this a little bit later, but because bacteria cause excessive immune responses, they can also stimulate our immune response to be detrimental to our own body cells. But why are viruses bad? Viruses turn cells into virus-making factories. So they co-opt or take over all of the cell's activities and make it so that the cell only focuses on making more virus. So the cell will not bother making energy. It won't care about making energy. It won't care about making its own proteins. It won't care about stabilizing anything having to do with itself, and just focus on making more virus. So this can be obviously detrimental to the cell because it's disregarding its own survival in order to make more virus. And in some cases, viruses actually destroy the cell when they get released. So some viruses don't have any way to interact with the cell transport system, so they just keep filling up the cytoplasm with virus until the cell membrane bursts, releasing the virus into the surrounding space. And so when that cell membrane bursts, obviously that cell is destroyed. So, how do we combat pathogens? Our first line of defense is our surface barriers. And the most important surface barrier we have is our skin. Our skin is the primary physical surface defense. It is what prevents bacteria from getting into our body. Our next surface barrier is the mucous membrane. This is found in orifices, so like the lining of your nostril, your mouth, your respiratory system, all have a mucous membrane, which is a layer of mucus that sits on top of the cells, and it acts as like a glue. It prevents any pathogen from getting into your cells. They get caught in the mucus instead. We also have some chemical barriers. The chemical barriers rely on chemical reactions to provide defense. So that would be things like acids, enzymes, mucin, defensins, and the lipids that we find in sebum and dermacidin. So if you would flip to your glossary, this is where I'm going to have you put your definitions. If you turn to page 5, look for where it says acids. And so the function of acids is that they inhibit bacterial growth. Then we have enzymes. Enzymes kill or break down microorganisms. Mucin is the sticky part of mucus. So this is the reason that the mucous membrane traps pathogens, because mucin is the sticky part of mucus that traps those pathogens. Then we have defensins. These are peptides that inhibit microbial growth. And then we have the lipids in sebum and dermacidin. Those are toxic to bacteria. So that should be what you need out of the glossary. So let's move on to antigens. In order for our body to defend against an infection, we have to recognize it as an infectious agent. We have to recognize it as a pathogen. That identification comes from a specific fragment of the pathogen. That fragment is called an antigen. The antigen is the recognizable part of a pathogen that our immune system is able to identify. So if you go back to our glossary on page 3, you see antigen, and it is the identifiable part of a pathogen. We use those antigens to create this very special protein called antibodies. The antibodies are the thing that actually recognizes the antigen. That is the thing that's going to allow the immune system to take effect. And back on page 3, again, you can find antibody right under antigen, and it is the protein complex that identifies antigens. So what do we do once we have an antibody that has recognized an antigen? what happens? Well, this is where our immune system goes through and kind of attacks that material. And there are several different ways that we can attack that material. So we have neutralization. This is where the antibody surrounds the pathogen. So you can see the different, that Y-shaped antibody connecting to the different parts of the pathogen. And that prevents that pathogen from infecting a cell because it blocks the binding sites. So that's one possible option for our immune system, is neutralization. Another option is agglutination. This is where the antibodies stick to multiple different pathogens. So like you can see, this antibody right here is attached to this bacteria right here and this one here in the back. So it clumps them up together. And when it clumps them up together, it makes it a really large complex so that our immune system can recognize it. And then we can also precipitate, which is where we have the antigen molecules being attached to each other by antibodies. All three of these methods lead to our immune system going through phagocytosis. So once an antibody is recognizes an antigen, one of these three methods is utilized, and then we phagocytize it, which is cellular eating, where we extend the cell membrane around the object, pull it in, and dissolve it by digesting it. And if you go to your glossary on page 3, the last term there is phagocytosis, which is cellular eating, and it's used to digest pathogens. So, what is the process of phagocytosis? This is, like I said, the process of cellular eating. And this is the process where most antigens are going to be broken down by. This process is carried out by neutrophils, macrophages, and monocytes. Macrophages and monocytes are actually the same thing. It's just where they're located that differentiates them. But you can go ahead and list them separately in your notes. And if we go and back to our lovely glossary, on the first page we have monocytes. Monocytes are the phagocyte that phagocytizes pathogens, and they are kidney-shaped. They have a kidney-shaped nucleus, and they are an agranulocyte. They are specifically found free-floating in the blood. Macrophages, on the other hand, are a phagocyte that phagocytizes pathogens, but those are found in tissues. They're not circulating in the blood. These are found in your skin or in your organs. They're found in functional tissues. Neutrophils are also on that page up at the top. It's the first one there. So neutrophils are going to fight bacterial and fungal infections, and they're going to do that through phagocytosis. They have a multi-lobed nucleus, and they are a granulocyte. So heading back to our notes, working through the stages. So first we're going to adhere to the pathogen. Then we're going to extend our cell membrane through the use of pseudopods. They look like little feet, so that's how they got their name, pseudopods. And they eventually will engulf, completely surround the pathogen. And they will form a phagosome, which is a phagocytotic vesicle, which will combine with a lysosome, and that lysosome will release acids and enzymes to start dissolving away the pathogen. Any toxic compounds are going to be kept inside the lysosome, and then the lysosome is going to expel out through exocytosis those waste products. So you might have noticed up on our chart that we had one other pathway, the complement system. And I didn't really touch on it because I was going to hit it more here. This is the way in which we regulate our own internal cells to trigger apoptosis. So this is one of the ways that we can cause a cell to go through the death process to actually self-destruct. So the complement system is how we trigger apoptosis. The interferon system is where we have interferon released by a cell to prevent infectious agents from being able to bond to that cell. So before a cell dies, one of the things that it can do is trigger the release of interferons, and those interferons go bond to surrounding cells, and those surrounding cells then have a heads up that infectious agents are trying to bond to them, and it prevents them from bonding. So it prevents them from bonding to a second host cell. Some other innate defenses we have: inflammation. Inflammation is the name for increased blood flow to an area. So we can dilate our blood vessels. We can have more white blood cells, things like neutrophils, flood into an area. And that can help respond to any bacteria or viruses that might be in an area. We also have fever. So fever is where we system-wide, body-wide, increase the body temperature so that we increase the survival. We decrease the survival of bacteria. So most bacteria want to be at our body temperature, but if we increase the temperature, we can cause them to die because they can't survive at that temperature. Now, the problem with fever is that our own body cells don't like to be at an increased temperature. We want to be at body temperature as well. We don't want to be at an increased body temperature. That can cause our own cells to be damaged. So when we talked about bacteria being problematic because, they can cause an immune response, this is what I was talking about. Having a fever can be very dangerous because it can kill off our own cells. high fevers can cause brain damage. They can kill off our neurons. So we don't necessarily want to have a fever for an extended period of time because it can cause damage to us. So just so you kind of know where we're going with things, we just talked about the innate defenses. So we just looked at surface barriers and our internal defenses, and we're going to be talking about our adaptive defenses next. But if you will turn quickly to the glossary, if you will turn to page 4, the complement system, the function there is the process where antibodies activate a cascade reaction that ends in cell lysis. Lysis, right underneath it, is the breaking apart of cells. Interferon, right beneath that, is the chemical messengers that prevent infection of surrounding cells. Inflammation is increased blood flow to an area, and it allows for more immune cells to work in an area. Fever is the systematic increase of body temperature. And that will take care of the last of our glossary items for this. So we will next hit our adaptive defenses in our next lecture.